Welcome to the project Online Analytics for Host Pathogen Protein Interactions Analysis. In this project, the host used are mouse and pathogen refers to the influenza A virus, otherwise known as IAV. A reliable analytical tool requires dependable data. Therefore, the goal of this project was to combine results from multiple similar scientific studies carried out in mice to perform a meta-analysis to deliver accurate and reliable insights on the virulence factors responsible for IAV infections. So, why is the study on virulence factors of IAV infection essential? Firstly, IAV is a significant danger to global human health and life, and it is critical to have a deeper knowledge of the virulence factors responsible for IAV infections to counteract potential outbreaks. You may be familiar with 2009 pandemic caused by swine origin influenza H1N1. From the statistics, it resulted in between 151,700 and 575,400 deaths in the human population worldwide. Pandemics like this often cause increased morbidity with vast clinical and economic impact. So, what is IAV? Here, we see the structure of an IAV genome. The combination of the HA hemagglutinin and NA neuraminidase determines the strain of IAV. Currently, 18 HA and 11 NA subtypes has been discovered. The virulence level of different IAV strains depend on the combination of HA and NA. Next, why are mouse models used? Firstly, mice are commonly used as the host in many individual experiments of IAV. As they are biologically very similar to humans, they are cost-effective and also easy to handle. Next, I will share about the materials and methods used in this project. This project starts off with data collection of IAV infection records from numerous literature searches. Data collected are then curated. Next, to construct the graph nodes, statistical host pathogen interaction potential between pairs of IAV mouse domains were calculated using this plot. Lastly, a GUI was implemented to present all the collected data, and network of interacting IAV mouse protein domains as a network of nodes was constructed. Tools used in this project includes the Universal Protein Resource UniProt knowledge base used to collect amino acid sequences of IAV proteins, and the UniProt ID of each protein was also noted. The UniProt proteomes was also used to collect protein sets from fully sequenced mouse genome. Next, the domain interaction statistical potential DISPOT tool was used to estimate the potential of an interaction between a pair of protein domains, given their superfamily. This GUI was created using Python library dash. To begin, users can select an IAV subtype, for example the H1N1, followed by a corresponding pathogen, which in this case is the A. Puerto Rico 8 1934 pathogen. The information table will then show corresponding IAV infection records, namely the PubMed ID to denote which literature paper the record was from, followed by the mouse genome, the lethal dose 50, which is the amount of virus when injected all at once causes death to half of the group of test animals. It is a way to evaluate the acute toxicity of a substance. And lastly, the virulence classification levels. This is the interacting IAV mouse protein domains as a network of nodes. The pink nodes represents the virus protein segments, while the blue nodes represents mouse protein domains. Clicking on either nodes displays the respective node attributes. Clicking on the virus nodes displays a pop-up showing the protein sequence of each segment. This is unique to each pathogen and will come in useful for our future work. Clicking on an interaction edge will display the DISPOT statistical potential score. Here, we also display a list of non-interacting viral protein domains. Like in any project, facing limitations are inevitable. Limitations of this project includes, firstly, it is currently not possible to differentiate IAV genome across different pathogen strains by using only the protein domains. The reason for virulence levels to differ across mouse strains infected with the same IAV strains has not been uncovered. Lastly, there are still protein domains not discovered, hence it is not yet possible to ascertain whether those domains are involved in any interaction. To bridge the gap of this project, future work can include not using this pod as a standalone protein-protein interaction prediction tool, and furthermore, machine learning techniques can be adopted to conduct detailed studies on protein embedding and discover the correlation between the various amino acid sequences that have already been retrieved in this project. Thank you!